Hello, good morning everybody. So, where we finished last time, from there we basically try to continue, and is a different module, is module 6 now, and we have reached to lecture number 26. And after sodium and potassium, we will now bring you or give you some essence on the role of the two other metal ions. So, firstly 3 lectures will be devoted to magnesium 2 plus, and the remaining 2 will be on calcium 2 plus. So, here in this particular class, what we want to see is that a very important thing when our body or any other living system undergoes is the phosphate metabolism. And sometimes we find that this particular metabolism is also directly related to the signaling process of the different cell systems. So, definitely we will talk about the magnesium 2 dependent enzymes and kinases. So, this typical term how we define and how we can understand about the different kinases we will see. So, in our human body how the different amount of magnesium is getting distributed, as we have learned so far that how we can consider about the corresponding concentrations of sodium as well as potassium within the cell and outside the cell. That means, this concentration is also very much important in understanding their relative amount what is available within the cell and outside the cell, and how these important metal ions are particularly for magnesium 2 plus, because we do not know much about the magnesium chemistry, magnesium coordination chemistry, apart from its main group metal ion chemistry and the organic chemistry, starting with that of your important Grignard reagent. So, magnesium dependent enzymes in glycolysis processes and how we can introduce. So, we are just separating out this introduction of the phosphate anion or the removal of the phosphate anion. Here we will only consider for kinases and others the introduction of the phosphate groups. So, if we consider that apart from our the relative abundances what we have seen last time, that the carbon, hydrogen, oxygen and all these and the different metal ions. So, in terms of that, magnesium is the 4th most abundant metal ion which is available within the cells in terms of its per mole concentration. So, more than 30 enzymes so far has been discovered. So, it is very interesting that earlier we are talking about that we know about 300 or more or 500 enzymes based on zinc. We know that zinc is the most important and the most essential metal ion for our system, and nowadays we take zinc supplements, zinc food supplements and genes in terms of their different medicines. Now, if we just allow, because day by day we are just enriching our knowledge in terms of these enzymes, how they are fully characterized, whether you have identified the presence of the magnesium ion or not, and their different reactions and the functions. So, more than of these 300 enzymes, the cofactors of all these cases are required basically starting from the protein synthesis, very essential part of our survival, for our livelihood. Then the muscle and nerve functions, that means during that binding or during the movement of these magnesium based different biological molecules, we will find that the nerve signals can be transferred. Then 2 more important thing what we all the time we are bothered about. So, magnesium is important in controlling the blood glucose amount as well as the blood pressure. So, finally, also we will see that it is required, because these are the importances where we can think about, where we can think about their control, but what is the actual reactions, the direct involvement of the that magnesium ion, magnesium 2 plus for the different reactions. So, we will basically talk about 3 things. One is your energy production. Obviously, you will be knowing when I talk about the involvement of magnesium for energy production, definitely we are talking about the ATP related thing, because we know that the ATP is your corresponding energy currency. So, we can produce for its energy can be obtained from the system if we go for the hydrolysis, or can be consumed from other food material or the glucose material or the carbohydrate material for its production. Then whether you have the oxidative phosphorylation reaction. So, oxidative phosphorylation reactions, that basically also drives many reactions in our system, and the glycolysis, which is the most fundamental reaction for the glycogen synthesis and the glycogen degradation also. Apart from its major contribution for bone formation and the structural development, which is also needed for two other very important molecules like DNA and RNA. So, magnesium dependent DNA RNA synthesis we should be knowing, and also the synthesis of antioxidant glutathione in our body. So, this typical understanding for the molecular mechanism, because how they are working. So, we will not be able to go in detail of all these things, but we will focus our attention on the magnesium binding, magnesium interaction and magnesium related coordination chemistry, because we want to know about the initial distribution of the metal ions in the environment. That means, the magnesium 2 plus homeostasis is therefore important, because if your cell is not having the enough amount of magnesium for its all these activities, you will have some problem. So, what we see now is the different properties. So, not that the metal property, but the metal ion properties, which are very important. So, when you have the magnesium, because everything will be getting from our food source or any other sources, will be getting magnesium as the magnesium ion. So, 50% of the total amount is basically residing on bones and remaining within the cells. So, 50% of that again. So, 50% in the cell, again another 50% of that of the cytosolic magnesium 2 plus is in the bound form. So, you see how good is in terms of its corresponding coordination or the chelation to the diphosphate unit of your ATP molecule. So, that is the most important part of knowing the magnesium related coordination chemistry or biocoordination chemistry or the biological role of magnesium. We can find out that the phosphorus oxygen bond, two such phosphorus oxygen bonds from say ATP or ADP molecule. So, that phosphorus oxygen, that oxygen basically which is bound to already to phosphorus, can function very good amount of coordination. So, we know that phosphorus, the phosphine itself, when no oxygen is there, the triphenyl phosphine and all are very good ligands to the metal ions. So, similarly, the ATP should also be a very good ligand for this magnesium ion, and the remaining 50% along with K plus is bound to the ribosomes. So, the distribution basically we should have some very good idea about what are these distributions, because people will be talking about and people are doing research also on identification or the monitoring of the magnesium 2 plus ion. How in your cellular conditions, how your cellular cells basically, how you can detect the presence of magnesium 2 plus. If somebody says then okay, I know that the concentration is 0.5 millimolar concentration within the cell. So, how can you find out? So, there should be some technique, spectrophotometric or spectropolarimetric techniques which can be there, which can basically give you the corresponding informations about the total concentration of magnesium. But before that, already we have seen the two other metal ions, the monovalent metal ion like sodium ion as well as the potassium ion, and in our the last two classes of this module basically are devoted to calcium 2 plus. So, if we consider all of them, all four together, and we all know like your periodic table, the electronegativity values, the ionization potentials values, all these things, you can have also the table for ionic radius. So, these ionic radius what we get here is that sodium, potassium, magnesium and calcium. So, if we consider that how they are varying, you see if we consider simply the ionic radius of sodium plus, which is 0.95 compared to magnesium, it is only 0.65 angstrom. So, whatever you are getting due to the charge basically. So, due to the charge your size is less, and which is less than that of your sodium. So far whatever we have learned about the typical coordination chemistry or biocoordination chemistry with respect to sodium as well as the potassium, whether that can be applied to magnesium or not, we critically see this particular case also whether it will be possible to substitute some of the positions of magnesium, because we know the typical presence of magnesium is important for its actual function. If we substitute that magnesium by some other metal ion, say calcium or sodium, you will not be able to extract out that information or that particular reactivity. So, you see the smallest one having by positive charge 2 plus positive charge is very important. That is why its the hydrated radius is also very high, is 4.76 angstrom. So, is basically gather more number of water molecules. So, the smaller size is important, is not that it will just can go for the binding, it can function as a very good ionophore to that of your corresponding crown ethers or cryptids. But these two quantities, the smaller typical ionic radius and the hydrated radius, bigger hydrated radius. So, you can have also the ionic volume and the hydrated volume also. Hydrated volume is also important, hydrated radius as well as the volume, which is 453 angstroms. This is a very big amount. So, whenever they will be moving for cellular signals or going to the ATP side or going to some other enzyme side, we will find that this particular volume movement is important. So, you have to allow for that much space to move the hydrated magnesium 2 plus ion to carry that particular ion in terms of its that much big volume of 453 angstrom. Then the exchange rate is also not very high, but is also 10 to the power 5 order, and the transport number. So, all these fundamental quantities which we can tag around magnesium 2 plus are important in understanding these functions, because what we see the functions, we see the mechanisms, but we try to understand in terms of its corresponding reactivities or the properties in terms of the magnesium 2 plus ion. So, we have seen here that the hydrated volume is very big and is 4 times bigger than its ionic volume. So, always, so that is why the properties and all these things are important when you talk about the synthesis in the laboratory, the synthesis of say Grignard reagent, the methyl magnesium bromide or methyl magnesium iodide. So, Mg2. We can talk about the reactivity of that magnesium in terms of its metallic reactivity in the metallic condition with that of your Rx. But here, since you have already the ions, so never forget these are amides. So, we are talking all these in terms of the amides, the metal ions. So, what we see now that the magnesium 2 plus has a much slower exchange rate of water in its hydration sphere. So, since the charge is very high, so it will be tightly bound. So, the exchange rate will also not be very high, and it will give you the slower exchange rate. Now, if you see that already we have discussed, already we have seen, and we are able to understand that the diphosphate part. So, the diphosphate part of ATP molecule having 2 oxygen donors on 2 phosphorus ends. So, PO and PO or PO minus and O minus charges are there. So, either you can have a charged bidentate ligand having 2 minus negative charge or a neutral PO-PO coordination to the metal ion center, because the charge will ultimately be balanced. You can have the corresponding delocalization of all these charges. But we should know that the hard oxygen donors from the phosphate units are coming and binding to the hard magnesium center, which is smaller in size also. So, you have the typical catalytic pocket, and if the ATP binding motif is available over there in that catalytic pocket, and magnesium can come and bind to that particular pocket. Then other phosphoryl transferase enzymes. So, phosphate binding, activating, hydrolysis and transfer. So, those are your phosphoryl transferase enzymes. So, the phosphate group basically will be able to move from one point to other by the use of your magnesium 2 plus ion. So, when the magnesium 2 plus ion is there, now we can think of, we can go back to your typical inorganic chemistry or coordination chemistry books or classes, that what is your coordination number. If I ask a very simple question related to that example what I am giving, because you are from your school days you are studying the Grignard reagent. So, Grignard reagent we know, we have studied many reactions and all these things, but if I ask you a very simple reaction, not that in the solid state, if you are able to make it or synthesize it in the solid state, but in solution, when we will be doing some reaction with some substrate or some reagent, your Mg2. So, you have taken in some solvent, say ether we are usually taking, or THF, tetrahydrofuran we usually take. So, in that particular solution, what is the coordination number of magnesium 2 plus when we dissolve the Grignard reagent in that particular solvent. So, coordination number is always important. Whenever you think of a metal ion, whether the environment is a biological environment or a protein environment, or your metal ion is present in your test tube, in presence of water, in presence of other solvent like little bit complicated solvent like DMSO, because we have seen that the DMSO can give you some environment, some kind of corresponding typical environment which is very much similar to that of your protein environment. So, either 4 or 5. So, it can be either tetracoordinated or pentacoordinated. So, you must have the corresponding number of proteins donors or the ATP donor groups like oxygens of the phosphate units can be available and can come around your magnesium and it will bind to that particular magnesium center. So, coordination number is always important. So, in this particular class basically when you are a master in coordination chemistry, you always think in terms of the magnesium 2 plus as if you are saying that I am only seeing first the magnesium coordination chemistry. So, its charge is there and high charge density definitely as I told you compared to your sodium and potassium, it is giving a corresponding very useful Lewis acid character. So, whatever phosphate you just simply give any phosphate group. So, it is basically alkyl phosphate or phosphonates you can have. So, RO groups are there. RO can be your typical water molecule like that of your phosphoric acid. RO can be your R is your alkyl function for alcohol. It can be your glucose function or it can be your ATP also. So, you have the magnesium 2 plus and it will be interacting with the OH or O minus only. So, if some charge from the OH like your hydroxide ion, so the interaction is basically the typical hydroxide type of thing. So, one you can have the one coordination site can be occupied in this tetrahedral phosphorus. So, the phosphate is present is the tetrahedral one and is try to interact with the magnesium center, but that magnesium center already we have seen that it can have very many number of bound water molecules. So, those water molecules are there and at some point that water molecule having some different pK value compared to a free water molecule. So, it can go for deprotonation. So, if it goes for the deprotonation, if some base is also available in the protein pocket, quickly it can go for your hydroxide bound form. And that hydroxide basically when it is activated by the metal ion and it is under control of the phosphorus center also can now form a strong bond in the second case when you have the trigonal bipyramidal transition state. So, when you have the trigonal bipyramidal transition state, the original O minus can still be bound to your magnesium 2 plus center, but it can loosen up there such that finally it can transfer the already bound hydroxide group from the metal ion center to the phosphorus center and you have the movement of the umbrella on the reverse side. So, earlier you have this particular say and then it is moving in that direction. So, inversion is taking place around this phosphorus center. Again we are getting back the corresponding tetrahedral phosphorus. So, let us see quickly what are the enzymes. Those are basically dependent on magnesium 2 plus for their functions. In glycolysis we have 5 enzymes which are dependent. So, if you can have 10 or more number of these, at least 5 you can have for this particular reactions. Then 4 are there and these 4 if we tag them for the different reactivity patterns, but the basic reaction is your simple phosphate group reaction for hexokinase, phosphofructokinase, then phosphoglycerate kinase and the pyruvate kinase. So, all these cases they are all kinases. So, these kinases are basically responsible for the phosphate group transfer from one to the other. Then we will see also the enolase in all. So, in that means the double bond is there and all is there, that means the hydroxy group maybe is there already. Like keto-enol tautomerism we know from our school days. So, keto-enol. So, if you have a keto group as well as you have the double keto groups also that can go for the in-all formation, that tautomerization to in-all. Similarly, that all O is deprotonated, you get the enolase function. So, that can be stabilized basically, is not that form is formed, that form we are getting, but you have to stabilize through metal ion coordination. So, as we have seen that it is binding to your ATP and ADP molecules and that is why you can consider that they are basically involved in all the reactions where we see that ATP and ADP molecules are there. You cannot see that okay, we have ATP and ADP molecules only without magnesium, because in almost all cases you will find that the activation of the ATP molecules or the stabilization of that ATP molecules will require the presence of magnesium 2 plus. But in all these cases there is no such strong binding, unlike your transition metal ions like iron or copper. The binding of magnesium 2 plus to the enzymes and their corresponding polypeptide donor groups or the donor atom is relatively weak, and only the corresponding association constant or the corresponding formation constant of the corresponding complexes is also less, is only 10 to the power 5 mole inverse. Then we just simply confine to say that we can have the PGT in kinases. What is PGT? PGT is nothing but your phosphoryl group transfer reactions. But the mother nature's tool basically is the mother nature's weapon, rather you can say is tool or weapon for introducing the phosphoryl groups 2 is basically introduction, not the removal. In case of phosphatases, not in kinases, in phosphatases we will talk about the removal of those phosphate groups. So, wherefrom the PGT reactions are basically important to add and remove the phosphate groups to or from the cellular metabolites. When we are metabolizing glucose, some intermediates we get basically or some corresponding glycogen assimilation, we can have something where you have to go for the corresponding attachment of the phosphate groups, or sometimes the macromolecules like proteins can also be phosphorylated. So, this sort of reaction is catalyzed by many regulatory enzymes which can also give us the corresponding signals such as protein kinases, protein phosphatases, ATPases and GTPases. So, you see not only these are very important for these kinases reactions, but is also important for many enzymes to functions, because the proteins are having OH functions, either that OH is coming from the alcohol end or is coming from the phenol end, and you can have also the different dinucleotides. So, when you go for the phosphorylation reaction on proteins, it directly takes part in signaling process and it also activates the glycogenolysis, that means the hydrolysis, and inhibits sometimes also the glycogen synthesis via phosphorylation of the protein side chain. What is that side chain? It is the alcohol end, that is just now I told you that alcohol end of the serine amino acid residue. So, serine has the alcohol function, OH function is there, and that OH function can be phosphorylated. So, that is the basic idea. So, whenever we are talking that you have the serine function, whether it can be phosphorylated or not, that serine has a very useful functions in the enzymatic world also in absence of metal ions sometime. So, when our system is basically consuming these two together, glucose as well as the dioxygen molecule, the amount is one is 80 milligram and amount is 50 milliliter per minute. So, you see that our system is always we are supplying the fuel and we are supplying the oxidant. Your glucose is your fuel and O2 is your oxidant, and that is required for the phosphorylation of the hexokinase. So, hexokinase you see this particular group is coming in the pocket, basically is entering there. So, it is try to move there. So, what is moving there? So, that we will see in yeast hexokinase. Why, how people have studied all these things is very important, and people have studied all these nicely on some model system, model living system, which is your yeast. So, yeast hexokinase basically shows the glucose binding. So, glucose molecule is coming. So, you should have the bigger hexokinase unit, and when it is coming and then finally binding to that particular pocket. So, it is initially it can be open up and it can close also little bit when the glucose is entering, and there must be some conformational change before we think of the corresponding reaction, which is your phosphorylation reaction. So, this phosphorylation reaction can occur from the inversion configuration around the phosphorus center. We have also seen is now is another typical example I am showing you. You can have alcohol, you can have water also, because that the very basic coordination environment around the phosphorus, which is important, is your trigonal bipyramidal environment. So, your glucose moiety is coming with your CH2OH, like your serine can attack your phosphate unit, and the phosphate unit and all oxygens are substituted by your the different types of oxygen centers. So, that basically gives us a different character of these, and we can find out the inversion process also. So, what we are doing? We are going for the glucose 6 phosphate. How quickly and how easily that can be phosphorylated by your ATP molecule. So, an ADP bound to one phosphate, it is nothing but your ATP molecule. So, it is basically the transferring. So, this basically the glucose is taking up that. So, already we have the ATP energy rich molecule in our hand. So, the two apexes, not small p, capital P center, basically the phosphorus center is occupied by the incoming and leaving group. So, one is your incoming group and another is your leaving group. So, glucose is coming and this ADP is moving basically as from the transferring one phosphate unit to you. So, you are getting a ternary complex out of that when you have the interaction of the magnesium 2 plus with your ATP molecule. So, what we see now that the protein domain closure. So, glucose is entering. So, you will have the protein domain closure places the ATP near the C6OH function of the glucose. So, you have that. So, how you attacking, that is why the end these two phosphate units are bound to the magnesium. So, it is basically binding through a water molecule to the carboxylate of a well conserved aspartate residue. So, aspartate residue is there, and that aspartate residue is important, which acts as a base to deprotonate the hydroxyl function on the sugar group. So, sugar or the glucose unit you have. So, if you have the aspartate residue, if we can deprotonate that unit. So, instead of OH it is O minus, which will be required for your phosphorylation reaction. So, the kinase phosphorylate the metabolites and the protein kinases phosphorylate the serine unit, threonine unit or tyrosine residue. So, all are having OH function, either alcoholic OH or phenolic OH. So, those are basically phosphorylated, there is no problem. So, only you have to find out which amino acid in the protein chain can supply you the OH function, and that OH functions can be phosphorylated. So, not that many other important molecules in this COVID time, we know all about cytokines. So, cytokines are also can be related to this particular one, hormones are there, because all these are important. Several growth factors are for protein kinase signaling nodes, because we have to go for the signaling process such that these hormones, these cytokines and all can be activated and give some important property or the function. So, these protein kinases have important roles also for in several cases the disease state, which makes them important targets basically for effective drug discovery. So, if you consider that we can stop the function or we can enhance the activity of these protein kinases by something, by some molecule which can go and inhibit these functions, we can have something which will be developed as a drug molecule. So, this phosphorylated letting the kinase structure interacts with the substrates. How? We have to know about the molecular contacts between the substrates and the active site residues and the different cofactors. That is a very important thing. So, three dimensionally from these particular structures, which is basically the visualizations. You can close your eyes and try to visualize your thing that you have the magnesium, something is coming and binding to your magnesium, but above all you have the substrate molecule. So, big substrate molecule will also come, and where it will come in the active site center, and the different cofactors are also there. So, from a big diagram, what we try to understand, you see now calmly that you can have the left hand side you have the hinge, and hinge is basically taking care through some hydrogen bonding interactions on the nucleotide base. So, adenine part of the base, and on the lower part you have the catalytic loop. Catalytic loop is supplying some amino acid residues. So, side by side ASN, LYS and ASP. So, my request to all of you that whenever you see these figures, try to understand is what type of amino acids we are talking about. So, you should be knowing the corresponding abbreviated form of ASN, LYS and ASP. So, ASP we all know is the aspartate groups. So, that is why we have the aspartate acid group and the aspartate anion which can be phosphorylated. So, the aspartate when it is bound to that OH function, which is nothing but your phospho acceptor. So, aspartate group apart from the other 2 amino acid residues within the catalytic group is therefore important to channelize these things, because one magnesium at the top which is your magnesium 1 which is important, and apart from that you can have another magnesium 2 which can have some role, but is not clearly identified till date. So, the interactions not only with the phosphate anions can also stabilize this particular lysine residue which is a conserved residue through only hydrogen bonding interaction. So, not only coordination, the dotted lines we are showing as the coordination bonds also. Sometimes if you have a very strong bond and at the bonding distance, the coordinate bonding distance is around say 1.9 or 2.1, we cannot consider these as the typical bond. So, these are the interactions like your hydrogen bonding interactions. So, it can be little bit stronger than hydrogen bond interactions, but it can be also weaker. So, because you just in a transient moment you go for these interactions and then the magnesium will be leaving that particular place once the reaction is achieved. So, the conclusion part, what we have seen, what we have noticed in this particular class is that we have studied about the kinases. You should be able to define what are kinases and how these kinases are important. Since the charges are there on the phosphorus oxygen bonds and the OR can be neutral with PO double bond, or sometimes we do not write as PO double bond, we write is a coordinate bond. So, because these are all basically when you determine the structure, they are having the phosphorus oxygen single bond distances. So, either the charged oxygen or the neutral oxygens or in between can give you the corresponding neutralization to your magnesium 2 plus center, and it can form or it can bind to that of your ATP complex. Then the different metabolic cycles, the kinases are important, the kinase enzymes are important, which are basically catalyzing the same reaction. We are talking all the time about the phosphate group transfer reactions. So, groups from ATP can be transferred to the glucose or any other carbohydrate molecule, can be transferred to the carboxylates like aspartate. Now, you can have the pyruvate. So, the pyruvic acid cycle we all know, and then the guanidine is a very important molecule, another type of molecule you can read it. I do not have that much time to talk about all these things, but what is guanidine and where we can get it. So, if you have creatine, so creatine factor is there in our system, so that creatine can also be phosphorylated. So, books are simply from the Wikipedia page, also the magnesium in biology and the Crichton's book also. Okay. So, thank you very much for your kind attention.